Okay, so today we will discuss about probabilistic models for biological sequence analysis. And biological sequences, DNA sequences and protein sequences, as you know, are sequences of symbols. For DNA, 4 symbols, A, T or C, G, and for protein, 20 symbols. So, people from many disciplines like mathematics, physics, statistics, they treat these molecules as symbolic sequences. And because of this nature of biological data, particularly DNA and protein data, it is easy to interpret and analyze them using probabilistic models. So, probability of observing these symbols or strings of these symbols, these patterns, so they can be modeled using probabilistic models. Thank you. Okay, so one of uh, the most challenging problems in 1980s in molecular biology and genetics was to identify genes in genomes. And genes, as you know, they code for proteins. Uh, Not all, uh, but those that are transcribed to messenger RNA, they could get translated to proteins. Of course, there are non-coding RNA genes that do not code for proteins. They do not make proteins. They can still make some structure that could be useful for certain functions. So, as I said, one of the problems in the field was to identify genes in genomes, and this problem was often called gene discovery problem, sometimes gene identification or gene recognition. And if you look at the genome of an organism, uh, it could be millions or billions of nucleotides, and it's really a challenge to identify these patterns, particularly in 1980s and 1990s, when methods were being developed for identifying genes. Nature has provided some hints actually, and those hints are, as you know, the start codons, the stop codons. So you know that the start codons in prokaryotic genomes are ATG, CTG, TTG, and sometimes I think GTG also. In eukaryotes, almost always ATG. And then when you read like this, first is ATG, start codon, and then next one, and then next one. And then when you encounter one of the three stop codons, TAA, TAG, and TGA, then those are signaling the end of the translation. So thus you have reached the end of a potential gene. Okay. So these triplets of nucleotides that start with one of these potential start codons and ends with one of these three stop codons, they are called open reading frames. Okay. And you would want to search for these sequences in the genome, open reading frames. But not all open reading frames code for proteins. Only a fraction of open reading frames code for proteins. Okay, for example, in E. coli, you can scan the genome and find about 40,000 or so open reading frames, maybe more. Okay, starting with one of the four triplets of nucleotides, ATG, CTG, GTG, and TTG, and ending with one of the three triplets of nucleotides, TA, TAG, and TAG and TGA. Okay, so those could be over 40,000, but only a fraction, just about 4,000 or so, code for proteins. So the gene finding problem is formulated as discriminating the protein coding open reading frames from non-coding open reading frames. Okay. So, of course, what nature has provided, that is the hints, they are used, but more has to be done. Otherwise, it won't be a computational problem. Okay. So, the first step in identifying genes is to scan all open reading frames, and I defined the open reading frame. Now you understand. Okay. Open reading frame starts with one of the four triplets of nucleotides ATG, CTG, GTG, TTG, and ends with any of these three. Okay. If an ORF codes for a protein, then that ORF is a gene, is a protein coding gene. Okay. Not all open reading frames code for proteins. So the problem is to identify those open reading frames that code for proteins. So here are the three steps in identification of genes. The first step is to find features, statistical features that can discriminate between genes and non-coding regions, non-coding sequences. Okay. So identify features, statistical features that can help discriminate between protein coding sequences and non-coding sequences. This is the first step. Once you have identified those features, then build a model for each of these sequence types. One type is protein coding, the other type is non-coding, as an example. Okay. So once you have built the models, the third step is to integrate these models into a pattern recognition algorithm that can help discriminate protein coding genes from non-coding sequences. Clear? The three steps. And we will go through all these steps in a moment. Okay, so from 1986, Mark Borodovsky, and that time in undivided USSR, he was a scientist in a university in Moscow, and he was examining the sequences of E. coli, coding sequences of E. coli, and there were only handful of such sequences, and this is the pattern that he observed. So frame 1 indicates codon position 1. You understand? So if I write a sequence, and you did actually homework also, and in the first homework I told you that the first nucleotide is located in codon position 1. Then you know that the second nucleotide is in codon position 2, the third nucleotide is in codon position 3, and so on. So that would be first codon and then second codon and so on. Okay. So frame 1 here means codon position 1. So just look at codon position 1 throughout the sequence. Okay. Just look at position 1 only. So 1, 2, 3, 1, 2, 3, 1, 2, 3, it would be like this, right? If there are n number of codons, you have to just look at position 1 and the nucleotides in this position across the sequence. Okay. So this is what they did, and codon position 1 here indicates frame 1, codon position 2, frame 2, position 3, frame 3. Okay. And they observed that, Borodovsky and colleagues, and they observed that there is a non-uniform pattern with respect to the codon positions. So you can see the probability distribution of these four nucleotides at position 1 differs from the probability distribution at position 2 and position 3. Okay. So you can observe more frequency of A or G at position 1 compared to position 3, for example. So more occurrences of nucleotide A or G at position 1 and C or T at position 3, or so on. So they started thinking about the biological regions for these patterns, and there could be manifold regions. For example, and this is what they thought, they investigated and they came up with some regions, and one of the regions was that coding sequences lack stop codons, right? coding sequences should only have codons that code for amino acids. And what are the stop codons? TA. So TAA, TAG, TGA. So they always start with T and at codon position 1, at position 1, right? So if you look at position 1, the frequency of T is much lower. So maybe that is contributing to that, many different reasons. And then they also observed that, and this I think actually was known much before, that there is an abundance of RNY codons in coding sequences, where R is purine, okay, purine, and purine is A or G. Y is pyrimidine, and pyrimidine is C or T, okay. And this is position 1, this is position 2, this is position 3. So at position 1, there is an abundance of A or G, okay, because of the abundance of R and Y codons in the coding sequences. So which is reflected here. The probability of A or G, add them up, is over 60%, right, at position 1. And the probability of C or T at position 3 is, so let's look at this, is about 55% or so, okay. So because of this region, the abundance of R and Y codons, you can see, you can correspond actually. So it could be the potential regions and why you are observing this pattern. Also, there could be manifold regions, as I said, it's really complex. You know the degeneracy of the genetic code. So some of the codons could be preferred for an amino acid. For example, GGU could be used 90% of the time, and rest, GGC, GGA, GGG could be used 10% of the time for encoding glycine, okay. So there would be more use because GGUs are used more frequently. So at the third position, you will have more U's or T's, right, in the coding sequence. So yeah, all these together contribute to the differential pattern that is reflected in the coding sequences, okay. On the other hand, if you consider non-coding open reading frames, and you can put the codon, even though they do not code for proteins, you can still put the positions. So first triplet of nucleotides 1 to 3, position 1 to 3, next 1 to 3, and so on, okay. And then obtain the probability distribution for the 3 frames. And it was done, and it was observed that the probability distributions at the 3 positions, they are more and less similar, okay. Similar and same as the probability distribution for the entire when the positions are ignored, okay. So which gives a hint that there is a pattern that discriminates protein coding sequences from non-coding sequences, okay. And these are which kind of patterns? Position specific nucleotide compositional pattern, okay. Position specific, frame specific, nucleotide compositional pattern, okay. So people started thinking about all types of features that they can consider for discriminating protein coding genes from non-coding genes, and one of those features was this. So this led to the development of algorithms. Many of those were based on Markov chain models. Markov chain models have been used frequently in the field of omics because of the reason I told you, that these are pluralistic models, and because of the ease of using these models for analysis of biopolymers, which are sequences of symbols, right? Those symbols could be the components that make these biopolymers. So because of these reasons, Markov chain models were adapted or adopted and were used in modeling of biological data. So let's look at what a Markov chain is, as mentioned on the slide. So Markov chains characterize stochastic processes. Stochastic means random processes. Random processes that incorporate a finite amount of memory. So it's not completely random. It is not memoryless. It does incorporate some amount of memory. Okay, good. A Markov chain model is used to describe systems that are governed by random processes with some amount of memory. Okay, so systems that can be described by such models that incorporate some amount of memory. Okay, so DNA and protein sequences may not follow Markov chain models. Okay, but if you consider these models, and because of the correlations that are there in these sequences, and correlations can arise for various regions, these models can capture those correlations and can discriminate. So far the task of discrimination is concerned, these models can do it, even though DNA and protein sequences may not be following exactly the Markov chain models. Okay, so the goal is to use the models to capture the patterns that can help in addressing a problem. And that Markov chain model did, and that's why it became so popular. Okay, so let's consider DNA sequence, which is CGG, AT, and so on, and last is GC. Okay, if each base occurs independent of the other, which means there is no memory, they occur independent of the other, they do not have memory of the previous base. So then the model is of order 0. The system has a memory 0. Okay, okay, very good. Finite memory, which means that each base occurs dependent on some previous bases. Okay, so there is some memory of the previous bases. So if the occurrence of a base depends on just previous base, for example, if occurrence of T depends on the just previous base A, there is a memory of how many nucleotides, how many bases? One base only. Yeah, sometimes I will say base or nucleotide means the same. Okay, so means memory 1 and Markov model of order 1. Okay, if the occurrence of a base depends on two previous bases, then the memory is 2, and this is Markov chain model of order 2, and so on. Okay, okay, so very good. If you understand this, then I can move ahead and I can let you know that Markov chain models were used for describing non-coding sequences and also protein coding sequences. So in the non-coding sequences, as I just showed you and told you, that the probability distribution of nucleotides across the 3 frames, they are more and less similar. Okay, so which means that you don't have to consider the codon positions. Of course, non-coding sequences do not code for proteins, so you can ignore the positions, and what you can do is to make a homogeneous Markov chain model of non-coding sequences. Homogeneous means not considering the positions. Okay, homogeneous with respect to the positions. So homogeneous Markov chain model of non-coding sequences. So frame independent or codon position independent. So let's consider this sequence. And if I ask you to compute the probability of this sequence for Markov chain model of order 0. So probability of this sequence given Markov chain model of order 0. Okay. So order 0 means that each of these nucleotides occur independent of each other. And you might have come across probability of A and B when A and B are independent of each other. Okay, so probability of A and B when A and B occur independent of each other is probability of A multiplied by probability of B. Okay, the multiplication rule for probability. Okay, so if each of these bases occur independent of each other, then the probability of S is probability of C. Yeah, same thing. Apply this. Probability of C multiplied by probability of G multiplied by probability of G, and so on. The last is probability of G. Okay. Markov chain model of order 1. Okay. So the occurrence of a base depends on the just previous base. So okay, so if I am here, okay, there is no previous base, right? So okay, so I am going to, so I can start in C and then I can move to G. So if I am here G, the occurrence of G depends on C. Okay. So I will start in C. So probability of C. So this is the initial probability. Probability of starting in C and then making a transition to G, and which is indicated by probability of G given C. Okay. And then G. So probability of G given G, and last and so on, and last is probability of G given G. Okay. So starting in C, so you can sometimes say starting in C, making transition to G, then making transition to G, and so on. Finally, in the end, making transition from G to G. Clear? Okay. Markov chain model of order 2. Order 2 means that dependence is on two previous bases. Okay. Okay, so two previous bases. So if I am here, no previous base. If I am here, only one previous base. So make a transition to this. Now you have two previous bases. Okay. So the probability of starting in CG, okay, probability of starting in CG, dinucleotide CG, and then making a transition to G. So probability of starting in CG and then making a transition to G, which would be indicated by probability of G given CG, and then probability of A given GG, and finally, and so on, and finally probability of G given TG. Okay, so you can thus model a non-coding sequence using a homogeneous Markov chain model. Okay. Okay, so okay, good. Yeah, some details I am skipping because of the time constraint we have. But I would like to see that you are still appreciating these mathematical concepts that were used for describing biological sequences. So protein coding sequences, they are made up of codons. And when there are codons, then you can visualize the codon positions, 3 codon positions. Okay. So which would indicate, and we saw that the probability distributions at 3 codon positions, they differ from each other. Okay. Which means that there are some biological regions why they differ, and you would want to capture this information, this biological information within the model framework. And that model framework is an inhomogeneous Markov chain model. Inhomogeneous means codon position specific or frame specific Markov chain model. Okay. Okay, so let's consider protein coding sequence modeling using inhomogeneous Markov chain model. So again, the sequence is the same, CGG, ATGG. Okay. What you don't know whether C is situated in codon position 1 or 2 or 3. Okay. So you would want to consider all possibilities. C could be situated in codon position 1. So the next would be position 2, next position 3, 1, 2, 3, 1, 2, 3. There are only 3 positions because codon is made up of 3 nucleotides only. Okay. The other scenario could be that C is situated in codon position 2. So what would be the next position? 3. So 3, and then next would be 1. So cyclic 1, 2, 3, 1. So 2, 3, then 1 will come 1, 2, 3, 1, 2. Okay. And the third scenario is C is situated in codon position 3. So next is 1, and then 2, 3, 1, 2, 3, and so on. Okay. So considering all these three scenarios, three possibilities, we can now compute the probability of this sequence to be a gene or to be a protein coding sequence in frame 1. So if frame 1, then you have to consider this. Okay, this is frame 1, codon position 1 for the first nucleotide. Okay. Okay. So let's consider this and compute the probability of this sequence to be protein coding in frame 1 given model order 0. Okay. So model order 0 means probability of C multiplied by probability of G multiplied by probability of G and so on, but now you have to keep track of position also. Okay. So you will put the positions also, which are shown as superscript. Okay. So probability of C position 1, probability of G position 2, and so on. Okay. Similarly, if you have to compute the probability of this sequence to be protein coding in frame 2 for Markov chain model of order 0, so you are considering this. So probability of C in position 2, probability of G in position 3, and so on. Just multiply these probabilities. Okay. And then let's consider the probability of this sequence, frame 1 sequence, under Markov chain model of order 2. Okay. Okay. So, and that means that Markov chain model of order 2, two previous bases. So you are basically considering trinucleotides, right? So 2 previous and the next one, trinucleotides. So, and then because of that reason, you have to keep track of the frame. So yeah, I can, is there something to rub this off? This thing, oh, okay, thanks. Okay, so okay, let's consider, where did I keep it? Oh, here it is. Okay, so let's consider, yeah, frame 1, this one. So CGG, okay. So all triplets of nucleotides, okay. So we consider the model order 2. So 2 previous bases and the next base. So 3 nucleotides. So CGG, and the next would be GGA, and then the next would be GAT. So GAT, right, and so on. Okay. So this is, so let's consider the reference position as the codon position for the first nucleotide of the oligonucleotide. Okay. So this is 1, 2, 3, frame 1, 1, 2, 3, and this is 2, 3, 1, and this one is 3, 1, 2, right? Okay. So and when we will compute, then we will put the superscript from the reference position that we get from the oligonucleotides. Okay. Okay. So you did it for non-coding sequence. Okay. You did it for that, and now we are going to do same now for the protein coding sequence, assuming that this is a protein coding. What is the probability of this sequence to be protein coding in frame 1 under Markov chain model of order 2? Okay. Okay. So same thing. So probability of starting in CG, and what is the reference position? Probability of starting in CG. So what is the reference? So you are considering this oligonucleotide. So what is the reference position? No, reference position 1. So that is why put a hat 1 here, that is indicating the position, reference position. and then making a transition to G. So then probability of G given Cg, right? Probability of G given Cg, and what is the reference position? 1, right? You are still considering this oligonucleotide, this trinucleotide. So reference position is 1, okay? So reference position 1, and then making a transition to A. So probability of A given Cg, okay? Probability of A given Cg. So what is the reference position? A given Gg, right? Oh, Cg, okay. Did I CGG? Probability of starting in CG, and then probability of given CG, yes. Okay, I think it is okay. What happened?
Sir, I'm not able to understand the last one, how we position
Which one?
frame one model order two.
Oh, order 2. How we are getting this?
So the positions one, one,
Oh, 1, 1, 2. Okay, okay. Let's do this again. So yeah, let's consider the this sequence, which is CGG. I will write the full sequence: CGG, ATGG, ATGG. Okay. Okay. So Markov chain model of order 2. So basically, we are considering triplets of nucleotides. So 2 previous bases and the next base, right? So, and I am going to consider all oligonucleotides of length 3. So, and this we already know 1, 2, 3, 1, 2, 3, 1. Okay. So, the first one is CG. So, can I get a better yeah, right. Okay, so the first is CGG. Okay, so 1, 2, 3, and I will say that the reference position, because there are 3 positions for this oligonucleotide. I will say that the reference position for this oligonucleotide is the position of the first nucleotide. Okay, so reference position is 1. Okay, good. And the next one is the next, so move by 1 nucleotide. So the next oligonucleotide is GGA. Okay, and this is 2, 3, 1. Okay, so reference position for this is 2. So the next oligonucleotide would be GAT, and then this would be 3, 1, 2, and so on. Okay, okay. So if I have to compute the probability of this with Markov chain model of order, this is a sequence of order 2. So I will write probability of S is equal to, okay, so if I am here, no previous nucleotide, I am here just 1. Okay, so I have to come here to see 2 previous nucleotides. So I will say probability starting in CG. So probability of, I will write first and then I will put the reference positions. Okay, probability of starting in CG and then transitioning to G. So probability of G given CG, and then next is probability of A given GG, and then probability of T given GA, and then probability of G given AT, and finally probability of G given TG. Okay. Okay. So what is the reference position for this? 1. What is the reference position for this? What is the oligonucleotide that this represents? Which oligonucleotide this is? No, no, no. What trinucleotide is this? No, I'm not saying position. I'm saying What trinucleotide is this one? CGG. Okay, so CGG. What is the reference position for this? Now you say 1. Okay, so 1. Okay, what trinucleotide this represents? GGA. So GGA position 2. Position? No, that's fine. Now position 3. So, okay, done. Okay. Okay, so this is how the non-coding sequences and protein coding sequences were modeled using Markov chain models. Okay. Okay, so now once you have the modeling part, which is actually computing the probabilities accomplished, then the other modeling part is that how you can compute the values of these probabilities. You need the values of probability of CG, this is the initial probability, and values of transition probability, probability of transitioning from CG to G, from GG to A, from GA to T, and so on. Okay? So you need values of these probabilities to compute the probability of the sequence to be protein coding in frame 1, right? And now we have ventured into machine learning. So you might have heard about machine learning, artificial intelligence, and all those things. So we are going into that. So we need to train Markov chain models to learn the values of these parameters. Okay? We want to train a model to learn the parameters of the model, and for that we need training data. And for this example, we need a data set of validated genes, validated protein coding sequences, and another data set of validated non-coding sequences. Okay? So train a model on validated protein coding sequences and train another model, Markov chain model, on validated non-coding sequences, or 2 models. Okay? So once you have trained, then you should have the values of these parameters, the initial probabilities and transition probabilities. Okay? And then you can compute the probability of a sequence to be protein coding or non-coding. So how to estimate the values of the parameters giving training data? So let's consider simple example. Let's consider the same sequence, and I am going to say that let's consider this a training sequence. Okay? This is a training sequence, and I want to compute the initial probability from, I want to estimate the initial probability or marginal probability of dinucleotide CG from this training data. Okay? So probability of this dinucleotide from this training data can be estimated from the counts of the dinucleotides. So how many total dinucleotides are there in this sequence? Okay? All possible dinucleotides. How many total? So CG, GG, GA, AT, TG, GG. So how many total? 6. Do it again. 6. You said 5? 6. 6. So L is the length. So 7 minus 1, 6. So of 6 of all dinucleotides, so total 6. How many of them are CG? 1. So the probability of CG is 1 by 6. So this is how model is being trained. So model estimated the probability of CG as 1 by 6. Okay? Now the transition probability. Probability of G given CG. Okay? Given CG means 2 previous bases are CG. So given CG means count of CG in the training sequence. So count of trinucleotides where first 2 nucleotides are CG followed by any nucleotides. Okay? So that is basically counting CG in the training sequence. So of them, how many instances are there that CG is followed by G? No, not now. Not so I am saying that this is the probability of G given CG. Okay? Given CG, how many of them have G following CG. That proportion is the estimate of probability of G given CG. Okay? Now you tell me that how many CGs are there in the training sequence? 1. So denominator is 1. How many CGG are there in the training sequence? 1. So 1 by 1. So probability of G given CG is 1. So the value of this transition probability is 1. So this is how machine learning models, they learn the parameters. So given training sequence, they are labeled data training sequences. So in this example, validated protein coding sequences and validated non-coding sequences. So train a model on validated protein coding sequences, and then train another model on validated non-coding sequences. So then you will have a trained protein coding model and a trained non-coding model, and then you can compute these probabilities, because now you know the values of these probabilistic parameters. Okay. So and this is important, because the question is that whether a given sequence is a gene or not. So you want to compute the probability of that sequence to be a gene, and you are considering all possibilities. So the probability of that sequence to be gene in frame 1, then in frame 2, then in frame 3. So 3 probabilities you are going to compute, okay? And perhaps more when I ask you to go to the next step. So gene discovery, 3 steps. Go back, 3 steps, and we did first 2. Okay, we identified features that can discriminate protein coding sequences from non-coding sequences, and then we built statistical models for protein coding sequences and non-coding sequences. The third one we are going to come in a moment, to integrate all these models within a pattern recognition formula or algorithm. Okay, so we will come to this in a moment, but I would like you to appreciate the power of modeling in prediction. So there is a problem in biology which is called CpG island prediction problem. Okay, so in the human genome, C in dinucleotide CG can be chemically modified by methylation. You understand methylation? Methyl group CH3. So CH3 can get attached to C cytosine in CG. Okay, and when C is methylated, there is a chance that C can get mutated to T, thymine. And because of this region, CG dinucleotide is rarer in the human genome. However, near the start of the genes, the methylation is suppressed, and because of that region, there is a relatively more abundance of CG near the start of the genes. And when there is an abundance of CG in a region, that region is called CpG island. Okay. So one of the problems in molecular biology is to identify CpG islands in a genome, and Markov chain models have been used for predicting CpG islands. So you learnt about how to model these sequences. So I can skip this because this is for a first order Markov chain model to be used for computing the probability of a sequence. So I will skip this because you understand. Okay. So the training has been done. So the transition probabilities have been estimated, and this is for first order Markov chain model. Okay. So probability of a sequence depends on the just previous nucleotide or base. Okay. Okay, so let's look at the model of CpG island. So this is indicated by plus, and this is the model for non-CpG island indicated by minus. Okay, and these are transition probabilities. So probability of, so this is yes, I think right. Probability of A given A. This is probability of C given A, probability of G given A, probability of T given A. Okay. So all these should sum to 1, because given A you have only 4 possibilities, right? Tossing a coin, you have only 2 possibilities, head or tail. Add them up, the probability will be 1. Same thing here. So rho should sum to 1. So let's look at the probability of G following C. Okay. So probability of G given C, which is this one. So which is 0.274. Okay. So dinucleotide CG. So probability of G given C. Okay. That probability is 0.274 for the plus model, for CPG model, and you would expect that for the non-CPG model, which was trained on non-CPG sequences, this probability should be substantially less than what you are observing in the CPG model, right? And that is shown here. That probability in the non-CPG sequences is 0.078, substantially less. Okay. So models have been trained, and now if you are given a sequence, so we will try again. If you are given a sequence, and let's write a sequence and compute the probability of that sequence. So given a sequence, T, C, G, C, G, A, C, G, G. Okay, this is the sequence. Okay. You want to use these models, already trained models, to predict whether this sequence is a CPG island or not. Okay. So you would want to compute the probability of this sequence to be a CPG island. So for that you are going to use which model, plus or minus? Plus. Probability of this sequence to be CPG island. So you will use plus model. Okay. So first let's write the probability of this sequence using Markov chain model of order 1. Okay. So tell me what I should write here. Model order 1, probability of T, then transition probability of C given T, and then probability of G given C, and last probability of G given G. Okay. Okay, and then I will put plus as superscript over P. Okay. And then probability of the sequence to be non-CPG island, so minus, and it would be probability of S to be non-CPG island sequence. So what should I write? Probability of T. What should I have the hat here? Minus. So okay, I am putting a hat or superscript, whatever you say, minus, and then probability of C given T minus so on. Okay. Okay, and then I will divide these two, and when I will divide these two, actually I will write equal to here now. When I will divide these two equal to, then what is this called? If I divide the probability of S to be CPG sequence by probability to be non CPG sequence, then what this ratio is often called? Likelihood ratio. Right? Likelihood ratio. Okay, so likelihood ratio. So this is the likelihood, and I have divided this. Okay, and when I take the logarithm of this, then this becomes log likelihood ratio, and this is also called odds score. Okay. Okay, so if this is greater than 1, if this is greater than 1, then what will you infer? The sequence is a CPG sequence or non-CPG sequence? CPG sequence. Okay. And if you are taking logarithm, so if logarithm is greater than 0, if you consider log A score, so if log A score or log likelihood. Yeah, a score is log likelihood. Okay. So if you consider a logarithm of likelihood, then this would be greater than, no, likelihood greater than 1. Log 1 is what? 0. So greater than 0. Okay. Okay. So which means that if odds score is, so log likelihood score is greater than 0, then this sequence is predicted to be CPG sequence. Okay. Otherwise non-CPG sequence. And the idea is simple, that if this sequence will have tons of CGs, then there would be tons of values 0.274 in the numerator and tons of 0.074 multiplied with each other in the denominator, and this is much higher than this, and because of this region numerator becomes greater than the denominator. So this is the whole idea. Clear? Okay. So if you understand this, then I will just skip this complicated notations to describe this model. Okay, you don't have to. If you understand this, done. Don't have to worry about this. Okay, so these models were used for predicting genes. We are coming to the third step, which is to integrate these models within a pattern recognition algorithm that can discriminate protein coding sequences from non-coding sequences. Okay. So given a sequence, and this is what we already discussed. So given a sequence, the probability of that sequence to be protein coding in frame 1, frame 2 and frame 3 can be computed, something like this. We just did it, and this is for a first order Markov chain model. And the probability of the sequence to be non-coding can also be computed. Okay, this is homogeneous Markov chain model description, and these are non-homogeneous Markov chain model description. Okay. Okay. So once you have computed these probabilities, then you can plot them. Okay. And what researchers did actually earlier, that they moved a window over a genome and then computed the probability of the sequence within the window to be protein coding in one of the 3 reading frames to be the non-coding. So here is the genome, and a window was moved, and window was of length 96 nucleotides. Okay, window was moved and then slid every 12 nucleotides. Okay, so next, and then next it was moved, and the probability of the sequence within the window to be protein coding in one of these 3 reading frames. So frame 1 and then frame 2 and then frame 3, and then the probability to be non-coding were computed. Okay, and then they were plotted actually. So okay, so computed, and then these were integrated within a pattern recognition algorithm. Okay, that was the third step. So compute this and integrate all these models within a pattern recognition algorithm. And a popular such integrative model is Bayesian formalism. Have you heard about Bayes theorem? No? Okay. Oh, you have heard? Okay, good. Yeah. Bayes theorem is probability of x given y is probability of y given x, probability of x, and then sum over x, probability of y given x. Can I get a better mark? So all these are drying up because you can't see from there, right? So okay, I will show you that, don't worry. I have put all these things on the slide. So probability of Bayes theorem, probability of x given y is equal to probability of y given x multiplied by probability of x divided by probability of y given x multiplied by probability of x and sum over all x. Okay, Bayes theorem. So what it does, it computes the posterior probability of x given y. Okay. So, let us say that and again example with the DNA sequence. Let us say that I have a sequence, and we know that bacterial genomes are packed with coding sequences, right. 90%, about 90% of a bacterial genome is coding, only about 10% is non-coding, right. So, I will ask you to predict. I have a sequence, and I have not shown you the sequence. I will ask you to predict whether the sequence is protein coding or non-coding, and you will be tempted to predict coding because 90% of the I will tell you that it is bacterial. You will say that okay, I can win if I will say coding, because there is a 90% chance of winning, right. So, you would be tempted to say protein coding, because in bacterial genome 90% of the genome is protein coding, 10% only non-coding. Okay. Now, I will show you the sequence, I will give you the sequence. So, your earlier belief, actually 90%, may now change given the evidence provided to you. Okay. So, the probability before seeing the evidence is called prior probability, Px. Okay. This is prior probability. Y is evidence. Thank you. Y is the evidence, Y is the sequence given to you, Y is the evidence. So, once the evidence is given, so probability of, so X is sequence, so X is not sequence, Y is sequence, so Y is the evidence sequence, okay. X is model actually, and you said protein coding, so that is protein coding model, okay. Okay. So, probability of protein coding, you said okay, you are tempted to say 90% because of your belief, because of your prior knowledge about the bacterial genomes, you will say 90%. Now, the evidence is given to you, which is sequence. So, your belief will change, and this is called posterior probability. Probability of model or probability of protein coding given the evidence, which is the sequence. Now, your belief will change after having seen the evidence. That is why this is called posterior probability. Okay? Posterior means after having seen the evidence. Okay. So, this will change, and importantly, if you see the denominator, X is model. So, all models can get integrated through Bayesian theorem, right. In the denominator, it is summation over X, and I said X is model. Okay. So, how many models are here total? 1, 2, 3, 4. Okay. So, all these can be integrated within the Bayesian formalism, okay, Bayesian theorem, which is in the denominator. So, let us say that X is model and Y is sequence. Okay. So, then it would be probability of a model given the sequence, given the evidence, is equal to probability of sequence given model multiplied by probability of model, and sum over all models probability of sequence given model probability of model. Okay. And so, here it is. So, let us consider coding model. So, let us consider this one, coding model frame 1, and I would say frame I, where I is 1, 2 or 3. Okay. So, posterior probability of protein coding. So, protein coding in ith frame given this sequence, okay, is equal to, use this one is equal to probability of this sequence given the coding model in frame I multiplied by coding model in frame I. So, same thing actually, use this, and then sum over all the models, the 3 coding models and the 3 and the non-coding model. Okay. Do this. Okay. So, this will give you the posterior probability of the sequence to be protein coding in frame I, where I goes from 1 to 3, right, 3 frames. And then you can also compute the posterior probability of the sequence to be non-coding. And when you sum all these 4 probabilities, they should sum to 1. Okay. Just see, just add these up. So, these are 3, I goes from 1 to 3. So, first you would compute probability of coding subscript 1 given S, okay, I is equal to 1. Then you would compute probability of coding subscript 2 given S, okay, and then you would compute probability of posterior probability of coding subscript 3 given S, okay, and finally you have probability of posterior probability of non coding given as total 4. Add them up, you will see that it is 1, okay, numerator will be same as the denominator, okay. Where? Oh, J is indicating that this J goes from 1 to 3, okay. So, this is summation. So, first consider J is equal to 1, okay. So, J is equal to 1, then this becomes probability of S coding 1, okay, probability of S given J is 1 coding in frame 1, okay, and then multiplied by probability of coding in frame 1. So, and then next is, so this is, so yeah, so this summation is from here to here because there is no J here, okay, and then next is J is equal to 2, and this summation means that keep adding up as you move to next J, okay. So, add to this which I just mentioned, J is equal to 2. So, when you consider J is equal to 2, then this would be probability of S given coding subscript 2, which means frame 2, okay, multiplied by probability of coding subscript 2, okay, and then plus, now J is equal to, consider J is equal to 3, okay, plus probability of S given coding, given code subscript 3 multiplied by P coding subscript 3, okay, and so done all 3. So, goes from J to 3, and then you add to that the probability of S given non coding multiplied by probability of non coding, okay. So, total would be how many terms? 3, sorry, so in the denominator of this. So, when you expand this, when you are just adding, so you will have 3 terms and this would be the 4th. Add them up, this is the denominator, right? Clear? Okay. So, yeah, unfortunately, I am not able to, because of time, I am not able to do everything on board. But you would try to understand and ask question, otherwise I don't know whether you are understanding or not. It is just not possible. These are mathematical models, but I want you to appreciate, without this omics course is incomplete, okay. Omics is not just use of software tools or web servers. Omics goes deeper into mathematical modeling and development of algorithms, okay. Okay, so once these probabilities were computed, posterior probabilities, then they were plotted. So, a window was moved over a genome, and for each sequence in the genome, these probabilities were computed, and then those probabilities were plotted. So, this is frame 1. So, okay, so frame 1, and then next 2, frame 2, frame 3. Okay, let us consider this only. So, probabilities were plotted. So, probability go from 0 to 1. Okay. So, they were moved, and then at some position. So, this is also genome position. Okay. On x axis is genome position. Okay. So, at some position, I would say that at this position there is a gene, and that gene is located in frame 1. So, the probability of this window to be protein coding in frame 1 will be low or high. I say that at this location there is a gene. So, the probability of that window that contains this sequence at this location to be protein coding in frame 1 will be high or low? High, because here is a gene. So, let us say that that position is this. So, suddenly there would be a spike here, probability, okay? and then gene is several windows, right? Gene could be, this is a window of just 96 nucleotides, gene could be 1000 nucleotides. So, for several of these overlapping windows it will remain high and then it will drop. Okay? So, and then you will say that looking at this profile, you will say that okay, there is a gene at this location in the genome and that gene is situated in frame 1. Okay? So, very good. So, this is what was done. Yeah, try, please try, otherwise yeah, I will not be able to cover many of the things. I am trying to help you understand with as simple descriptions as I can make, but these are not as simple models. They have been used for annotating genomes. So, you can see that at this location, from this to this, from here, this position to this position, there is a gene and that gene is in frame 1 on direct strand of DNA molecule. Okay? So, there are 3 frames for direct strand and 3 frames for complementary strand, and one more frame would be for non-coding. Okay? Okay. So, what earlier people used to do, they were analyzing DNA strands in turn. So, first direct strand and then complementary strand. Okay? So, when you do for the direct strand, then you are computing these probabilities. So, 3 here, I goes from 1 to 3 and non-coding. Okay? And then you do for the sequence in the complementary strand. Okay? So, do in turn. So, when this was done, so first the probabilities were, posterior probabilities were plotted for the direct sequence and then for the complementary sequence. And what was observed, that the strand opposite to where a gene was earlier predicted, that location also had some signals. Okay? So, a gene was predicted here on the direct strand at this location. Opposite to this is a region on the complementary strand. Okay? And there is no, actually there is no gene there, but signals were observed, and that led to many false predictions, spurious predictions. So, there is a gene, and people said that, let us say gene shadow. So, there is a gene in this strand and there is another strand here. So, on the other strand, opposite to the gene, there was a prediction. Okay? So, in the gene shadow region there was a prediction because of these spurious signals that arose, and people started thinking why these signals are coming in the region opposite to, in the region of the strand opposite to where the gene is located. Okay? Why that is happening? Finally they were able to figure this out. They found, and as I said, that there is an abundance of R and Y codons, okay, in genes, in coding sequences. And let us complement this. So, this is on the direct strand 1, 2, 3. Okay. So, 5 prime to 3 prime will read like this. Okay. 1, 2, 3 is these are the codon positions, and on the complementary strand I would read in the opposite direction, 5 prime to 3 prime, but from the other direction. Okay. So, what is the complement of this? So, this is pyrimidine. Pyrimidine is C or T. So, what would be the complement of C or T? So, C G and T A, right? G A, and N is any nucleotide, so N N, and this is purine. Purine is A or G. So, what is the complement of this? C or T, right? So, this C T is Y, and this is N, and this is R, and this is 5 prime and this is 3 prime. Okay. I think I should throw these, otherwise it's confusing actually which one. Okay, so now you have to read in this direction. So then you will read R and Y. Okay, so in on the other strand there was again an abundance of R and Y codons. Okay, so on the strand where there was a gene, of course there was an abundance of R and Y codons. On the other strand opposite to where the gene is located, there also in the gene shadow region on the other strand there was an abundance of R and Y codons, because R and Y codons are self complementary. Got it? Complement and read from the other direction. That's again R and Y. So because of this pattern, there was a false signal on the other strand. So you can see the false signal. And this also happened because researchers were analyzing these strands in turn. So when the model has seen this, model has not seen this, or when the model has seen this, the model has not seen this. This is a very strong signal, and this is relatively a weak signal compared to this. Okay? But because model has not seen this, and if you remember, if you sum all these up, sum should be equal to 1. So model, for example, for the shadow region, model gets most of the strength from here and rest are almost 0. Okay? And that is why this gets most of the strength is close to probability 1, right, at most of the locations. But if you allow model to see both the strengths together at the same time, then it is possible that this signal will pale in comparison to this signal, because there would be 3 for this strand, and then 3 for the other strand, and then non-coding, right? So when the model has seen all, then and they all should sum to 1. So this gains most of the strength, because this is so strong, this will be close to 1, this will get to 0 for that region, right? So you have to show the model everything at the same time, and this is the beauty of machine learning. So do not do in pieces, let the model see everything so that model can take right decision, just as human can take a right decision when human is shown all the evidence at the same time, not in pieces. Okay? Yes. What? हो सकता है gene, but in bacterial genomes genes do not overlap like this, they overlap at the ends. So this is well known, and that's why people wondered that why this is so. So in the gene shadow of every gene there was this signal, and that's a false signal, because in bacterial genomes genes do not overlap fully, they do not overlap like this, they can overlap at the ends. Okay?
But in viruses it says the gene overlap like this.
Which one?
For viruses.
Viruses can overlap fully. Okay, that's another thing, but yeah, but here they were focused on the bacterial genomes, and in bacterial genomes 2 genes do not, on the 2 strands they do not fully overlap, they overlap at the ends, and this is something that has always been incorporated in the prediction models of bacterial or prokaryotic genomes. Often models assume that 2 genes do not overlap at all, earlier models. So there is at least one nucleotide between 2 genes that are on different strands. But now later models, they also included the modules for overlaps to predict overlapping regions in the genes. Okay, so if model has seen everything, then the model should be able to predict correctly the gene, right? It would be ordered to eliminate the spurious signals. So this is a spurious signals, it will just get discarded, because this is so weak compared to this. Earlier model was not shown this when model was computing this, and this was gaining most of the strength, sum should be equal to 1. So it showed like this compared with other weak signals, but when model is compared with this strong signal at the same time, it gains most of the strength is close to 1, this becomes 0. Okay? Yes. Yeah, this is for only prokaryotes. Eukaryotic model would be different from this, even though the idea will remain same, that you have to build a model for protein coding genes, and then so you will say model for exons, the coding sequences, and then a model for non-coding sequences, and model for introns or splice sites, and so on. So you will have more features that you have to consider for eukaryotic gene than for prokaryotic gene. So to help you understand this, I have considered the simplest model, that for the prokaryotic gene, to help you understand how models are used for prediction. Okay, so I think we discussed this. So now how many models will be there? 3 models for coding, 3 models for gene shadow of gene, right? Shadow of gene. So how many? 6, and 1 for non-coding, total 7. So total 7 models. So when 7 models were used, then of course you compute the posterior probabilities of these 7. So probability of sequence to be coding in one of the 3 frames, probability of the sequence to be shadow, gene shadow, again in one of the 3 frames, and non-coding. Okay. So when that was done, can you see the signal is false signal is gone? It's gone. Okay, and this program was named GeneMark, and was used in annotating the first bacterial genome that was sequenced in 1995. Do you remember the first bacterial genome that was sequenced? No, not E. coli. E. coli 1997. First bacterial genome sequenced in 1995. E. coli may be second, not first. No, not that. Not that. Have you heard about Haemophilus influenzae? Okay. It does not cause influenza. That's a virus actually. H. influenzae, Haemophilus influenzae. It's a shorter genome. I think 1.5 megabases or 1.7. You can check actually. So around that. So this program was used for finding genes in that genome. Okay, so and here is a posterior probability profile generated by GeneMark program. Actually, you can go to you can just Google search GeneMark. You can go there and you can copy paste a genome sequence, and it will provide you where the genes are located. Okay, so now that you understand, you should be able to. So let's look at the profile generated by GeneMark. So can you let me know how many genes are there in this sequence, which is starting at this position? Okay, let's say 0th position. Okay, from this position to this one, which is about 20,000 something. Okay, whatever it is. Yeah, 20,000 something. Yeah. Okay, that's fine. So how many genes are there in this sequence? Is this a gene? Looks like, right? Yeah, looks like this is a gene. So and they put a threshold here that the windows should have the posterior probability greater than 0.5 and many of them, right? So eventually they take average and then they ask whether it is better than or greater than the threshold or not. Okay. So there is a gene in which a strand, direct strand, okay, in frame 1. Very good. Now you understand. Yes. And these are noises. Okay. Again, this is a noise, it seems like. So second gene is in which strand? Complementary strand. Which frame? First frame. And then third gene is here. Which frame? Third frame. And fourth gene also in third frame. So thus you know actually where the genes are. So this is the output that program will provide you. Okay, run GeneMark, it will give you this, and then you can go through this profile and you can say that okay, there are 4 genes and they are located from this position to this position and they are in these frames. Okay. Now when you run GeneMark, then you should be able to interpret the output from GeneMark. Okay. So one of the challenges with the use of Markov chain model for predicting genes by GeneMark was that the predictions were made for windows. Okay. So the probabilities were computed for the sequences within the windows, and which means that the model has not seen the entirety of the information that is there in the entire sequence. It is seeing only the information that is contained within the window, and that is why they did heuristic. They tried many different window sizes. So longer window sizes, good, more information, but it reduces the resolution of finding the boundary between protein coding and non-coding sequences. Okay, if you increase, because the resolution is as good as the window size. If you as good means that, okay, if it is longer, then resolution is bad. Okay, if it is short, then you are getting close to the location where the boundary is between protein coding and non-coding. Okay, as short, if it is just one nucleotide resolution, that is the boundary, because boundary is just one nucleotide. Okay, if it is longer, then resolution gets worse. So that was a problem, and the other problem was that when you move a window, and let us say that this is the boundary, okay, and window is here to here, and this is the boundary. So a part of the window is protein coding and another part is non-coding, right? But the entirety of the window has to be predicted protein coding or non-coding. So there is another limitation, that a window that is spanning the boundary of protein coding and non-coding has both protein coding and non-coding parts, but the model is constrained to predict the entirety of the window to be protein coding and non-coding, right? So that's a problem. So to address that problem, people started thinking about new models, and one of the models, again based on Markov chain model, is called hidden Markov model. So hidden Markov model can consider the entirety of the information that is within a given sequence for predicting a nucleotide to be protein coding or non-coding, which means that the resolution is at the single nucleotide level, right? No window, entire sequence information, and you predict based on this entire information a nucleotide to be protein coding or non-coding. Okay, and because of that reason, the other limitation also gets addressed, where a window is spanning boundary between protein coding and non-coding, and entirety of the window has to be predicted either protein coding or non-coding. But with this model, hidden Markov model, you don't have to worry about this, because each base is now being predicted as protein coding or non-coding using the information that is there in the entirety of the sequence. Okay. So then you would wonder what is the hidden Markov model, and it has been frequently used in the field of omics, proteomics, transcriptomics, genomics, to name any. Yes. T. Okay. I am not keeping track of the time. Okay. So let's have a tea break, and we will come back. Okay. We will be back around 12. Okay. So yeah, tea break. We should come here around 12. Okay. We will continue, and then we will have lunch break, and we will come back around 2. Okay. So let's use the time so that you learn modeling today and do some tutorials also. Okay. 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 So hidden Markov models. Hidden Markov models define a class of probabilistic tools to describe a system with hidden parameters. Hidden means unknown. So there are known parameters, there are unknown parameters, but both are integrated together within one system, and that system is hidden Markov model. Okay. With the one that we were earlier discussing, we developed model from knowns and then we were predicting unknowns, right? Here both are integrated into one system. So an example here is this DNA sequence CGG, AT and so on, last is GC. What is known? The nucleotides, okay, what you observe. So sequence of nucleotides. What is unknown or hidden? Whether C is protein coding or non-coding, whether G is protein coding or non-coding. So the functional or structural characteristics of these residues or nucleotides, okay, they are not known to you. What is known is the sequence only. So both knowns and unknowns, they are integrated within one model that is called hidden Markov model, and both sequences known sequence and hidden sequence, they are characterized by Markov chain model. Okay. So let's consider some examples to understand this. So coin toss problem. So I have a coin and I am tossing it, and there is a curtain between me and you. So I have two coins and I am picking one of the two coins and tossing, and then I am showing you the result, whether it is head or tail, and you are writing. So what you are observing is the outcome, whether head or tail. You are not able to observe which of the two coins I picked up and then tossed. Okay. So coin identity is hidden from you. What you know is the outcomes for each coin toss. Okay. Okay. So here it is. Here is how you can model this. So there are two coins, coin number 1, coin number 2. They are being tossed and then the outcomes are being shown to you. Okay. And now I am going to ask you to model it. So there are two sequences. One is sequence of the outcomes that you know because you were recording. So first toss head, second toss tail, third toss head, fourth toss head, you are recording it. Okay. So this is a sequence of outcomes that is known to you. Okay. What is unknown is the sequence of identity of the coins that was picked and tossed. Okay. Each time. So maybe I picked coin number 1 first time and tossed. So I am also writing actually. So that is known to me, not to you. So I am writing 1. Second time again I picked coin number 1, so I will write 1 again. Third time I picked coin number 2, I will write 2, and so on. So this is a sequence of unknowns, because this is not shown to you. You are only shown the outcomes. So there are two sequences of knowns, sequences of unknowns. Both are characterized by hidden Markov models. Okay. So let's consider the sequence of unknowns, the hidden ones. Okay. So what are the parameters? So there are 2 coins. So Markov model parameters. Now you should be able to tell me. So there are 2 coins. So you know initial probability. Now you know transition probability. Okay. So it's very simple. Two states, state 1 which is indicating coin number 1, state 2 which is indicating coin number 2. So I am asking you to describe this using Markov chain model of order 1. So Markov chain model of order 1 means memory 1. Okay. So probability of choosing coin number 1 given that the previous choose was coin 1, or probability of choosing coin number 1 given that the previous selection was coin number 2, right? So only two possibilities. So so you would want to now define all these probabilities. So probability of transitioning from 1 to 1, okay? So probability of 1 given 1, probability of transitioning from 1 to 2, probability of 2 given 1, given that the previous pick was 0.1, okay? Probability of transitioning from 2 to 2, probability of picking 2 given that the previous pick was also 2, okay? And probability of transitioning from 0.2 to 0.1, okay? Probability of picking 0.1 given that the previous pick was 0.2. So all these probabilities are now indicated. This characterizes the sequence of unknowns, okay? And then a sequence of knowns, which are the head and tail actually that you are noting down. So a probability of, and that is zeroth order model, probability of getting head from coin number 1, okay, P1. So probability of getting tail would be 1 minus P1, okay. So they should sum to 1. From coin number 2, probability of getting head is P2. So probability of getting tail would be 1 minus P2. Okay, very good. So this is given to you, observed sequence of head and tail. This is shown to you. This is not shown to you, right? The sequence of coin identity, coin that was picked and then tossed, which coin was picked. So that sequence was not shown to you, that is unknown. Hidden Markov model will help you predict this, predict the unknown sequence given the known sequence and the model, okay? Okay, let's see how we will do this. Again, tossing 3 coins together and same thing. So when you have 3 coins that you are tossing, how many parameters will characterize this sequence using Markov chain model of order 1? How many parameters for Markov chain model of order 1? Okay, how many parameters were there for 2? For the sequence of unknowns only, how many? Only transition probability parameters. So how many? 1, 2, 3, 4. 4 parameters, transition probability parameters. So when there are 3 coins, how many parameters for 3 coins? 9. So you said 9? Right, 9 parameters. So you can see actually 9 parameters indicated here. So probability of transitioning from coin 1 to coin 1, from coin 1 to coin 2, from coin 1 to coin 3, and so on, right? So total number of transition probability parameters would be 9. And similarly, probability of, this is for the observation sequence. So probability of getting head and tail from coin 1, coin 2, coin 3, and so on. Again, same thing, you will be given the observation sequence or sequence of knowns. You are not given this, you have to predict this. Okay, this is not known to you, this has to be predicted, and hidden Markov model will help you that. Okay, so here are the elements of hidden Markov model. So there is an output alphabet A. For DNA sequence, output alphabets, output. So for DNA sequence, you can say that it could be A, T, C, G, that is observed, right? A state space, a hidden state space, which is hidden from you. So let's consider simple example of protein coding and non-coding, that is hidden from you, okay? Whether a nucleotide is protein coding or is non-coding, that is not known to you. So a hidden state space could be just protein coding and non-coding. So just remove these two for simplicity. Okay, you can have as many hidden states you want, but let's consider only two here: protein coding and non-coding. Okay, a probability of transitioning between states, between hidden states, and we just learned actually probability of transitioning between coin 1 to coin 2, coin 1 to coin 3, and so on, right? So probability of transitioning between hidden states, and hidden states here are coding and non-coding. So probability of transitioning from protein coding nucleotide to non-coding nucleotide, probability of transitioning from protein coding to protein coding, and so on, right? So that is the probability of transitioning from one state to another state. And next would be output probability distribution, probability of observing an outcome from a hidden state. So probability of observing A from protein coding state. Okay. So we will try to understand further as we go to the next slides. Here it is actually probability of, let's see this one, probability of getting head from state 1, coin 1, okay, probability of getting tail from state 1, coin 1. So this is output probability distribution. Okay, P1 and 1 minus P1 is output probability distribution from which state? From which state? 1. Okay. Similarly, output probability distribution from state 2. Okay, clear? Okay. So that's it actually. This is this. And then initial state distribution, probability of starting in a hidden state. Okay. So for example, probability of starting in protein coding state and then emitting a protein coding nucleotide. So hidden Markov model is often interpreted as emitter of symbols. Okay. So there are states, hidden states, and from each hidden state, symbols will be emitted. So if it is a protein coding state, it will emit these nucleotides A, T, C, or G from that state. If it is non-coding state, it will again emit these symbols, these nucleotides from the non-coding state. So this is how hidden Markov model is often interpreted. So of course, nothing is emitted here. What is important that you should understand the model. Model is being explained as emitter of symbols. So that should help you understand. So the probability of observing a sequence O1 to OK is given by this. So don't worry at the moment. I will explain in a moment. So so let's consider two sequence. The hidden sequence and observation sequence. So okay, so I will use the same notation, and I think it's not difficult to understand. So probability of hidden sequence is S, observation sequence is O. Okay, this is the joint probability of hidden sequence and observation sequence. So this is probability of, so I'm just factorizing it, probability of S multiplied by probability of O given S. Okay. So joint probability is equal to marginal probability multiplied by conditional probability. Okay. Will not go into everything, but just you can explore this further. Yeah, this is it. So joint probability can be written as a product of marginal probability and conditional probability. Okay. So let's consider PS. PS is the sequence of hidden states, and that is S1, S2, Sk. Okay. because it is k symbols long. Okay, and I am asking you to compute this using Markov chain model of order 1. So what should come first? So this is capital S, hidden sequence. So probability of this sequence given Markov model of order 1. So what should I write here? P is starting in S1 and then transitioning to the S2 given, and last is P Sk given Ak minus 1. Okay, very good. So first order model. Okay, so I wrote for this, and then probability of O given S, probability of observation sequence given the hidden sequence, and 0th order model. Okay, so an observation sequence I would write like this: O1, O2. Okay, so DNA sequence O1, O2, and last is OK. Okay, and this is conditioned on this, right? Conditioned on the hidden state. So that's why I say that, okay, let's say that S1 is emitting O1 and then making a transition to S2, then emitting O2. Okay, and then a transition is made from Sk minus 1 to Sk after Sk minus 1 has omitted Ok minus 1. Okay, and then finally Sk is emitting Ok. Okay, okay. So so now how I should write this using 0th order model? So probability of, yes, probability of,
O given S when S is basically known.
no, S is unknown, S is hidden state.
Given S, what is that?
Even S is, it is conditioned on S. So S is emitting O. Okay, so this outcome is dependent on this state. Okay, that is it. So probability of 0th order model. So 0th order model. So probability of O1 given S1. Okay, so next would be probability of 0th order model O2 given S2, and last would be probability of OK given Sk. Okay. Now multiply these two, because you want to get the joint probability of hidden sequence and observation sequence. So multiply these two, and which is shown here. When you multiply these two and rearrange, and P is indicating the probability of transitioning between hidden states, and Q is indicating the output probabilities. Okay, both are probabilities. Okay, so if you will rearrange, you write it, and if you will rearrange, then it is something like that: probability of starting in S1, okay, and then emitting O1, which is here. Probability of emitting O1 from S1. Okay. Then what happens? A transition is made to S2. So probability of transitioning, there is a mistake. Probability of transitioning to S2 from S1. So this should be S1. You can correct it. Okay. So once you are in S2, then what happens? O2 is emitted. So probability of emitting O2 from S2. So O2 given S2. Okay, and you can do like this, and when you come here, so this is basically probability of transitioning from Sk minus 1 to Sk, and then emitting OK from Sk, right? Probability of OK given Sk. Done. So you just rearrange that what I showed you, you will get this. Okay, and this is the joint probability of hidden sequence and observation sequence. Okay, and this is what is being shown here actually, only this portion shown here. So if you sum over, and actually don't worry, I will skip that. So yeah, just I just discuss this, and don't worry about this, because I'm not going to come to this today. So this thing clear to everyone? Okay. So if you understand this, then you are understanding hidden Markov model. So what we want to do, we want to use this to predict a splice site in a gene from an eukaryote. Okay. So before that, actually, we would like to learn about how hidden Markov models have been used for describing prokaryotic genomes. So one of the most used hidden Markov models, the classical hidden Markov model or standard hidden Markov model, is one that emits one symbol from a hidden state. Okay. So let's consider this hidden Markov model of a prokaryotic genome, bacterial genome, where the states are shown in ovals. The observation symbols are shown in squares. Okay. So this is these three ones are coding states, and these three are the shadow states, and you can interpret this as gene on the direct strand and gene on the complementary strand. Okay. So and this is the non-coding state. So again, let's interpret this as hidden Markov model as an emitter of symbols. So if you start in a non-coding state, then a non-coding nucleotide is emitted. Okay. And then a self transition can be made to non-coding state, emitting again a non-coding nucleotide, because often non-coding regions could have multiple nucleotides, right? And then a transition could be made to gene on the direct strand or gene on the complementary strand. If a transition is made to gene states or coding states on the direct strand, then a codon will be emitted. So this is codon state 1, 2, 3. So there are 3 positions. So they are here. So it will go one by one, emitting the nucleotides of the codon. So the first codon will be emitted, and then a transition is made back to the first state of the codon, and then 1, 2, 3 again, second codon will be emitted. Once enough number of codons are emitted, then a transition will be made to the non-coding state, emitting non-coding nucleotides. Okay. And if you happen to come here, actually, so these arrows have associated probabilities. So if you happen to come here, the the codons from genes on the complementary strand will be generated. Okay. So yes.
What exactly do you mean by
Oh, emitted. Yeah, emitted is a hidden Markov models are easy to understand when you interpret them as states that emit symbols. Okay. So the reason actually we say like this because these models are generative models. You might have heard about artificial intelligence model, generative models. So they generate sequences, which means they emit sequences, they create sequences, right? So DNA and protein sequences have been created by nature, right? But in order to understand them, we use models, and models are simplifications. Models can generate these sequences, can emit these symbols or units of the polymers. Okay, those are artificial sequences, but they can mimic natural DNA and protein sequences. Okay. So that's why when people are doing modeling, they say that we generate DNA sequences, we generate protein sequences. The model emits nucleotides, the model emits amino acids, resulting in generation of protein sequences or DNA sequences. Clear? Okay. So a standard hidden Markov model emits one symbol from a hidden state. Okay. The generalized hidden Markov model of bacterial genome can emit more than one or more symbols. Okay. For example, again, the ovals are the hidden states. So if you happen to start in non-coding state, then a non-coding sequence will be emitted. Many nucleotides of the because non-coding sequence could typically be multiple nucleotides. So non-coding nucleotides will be emitted, multiple non-coding nucleotides will be emitted according to which model of non-coding sequence? You just learned. Non-coding nucleotides will be mated according to homogeneous or inhomogeneous Markov chain. Homogeneous, non-coding. Inhomogeneous for protein coding. Okay. So homogeneous Markov chain model of non-coding sequences, and according to the probability distribution of non-coding sequences, of probability distribution of the size of non-coding sequences. Okay. And then a transition will be made either on the gene state on the direct strand or gene state on the complementary strand. So if a transition is made to a start state on the direct strand, then a start codon will be emitted, and then a transition will be made with probability 1 to the coding state emitting coding sequence according to which model, inhomogeneous or homogeneous Markov model of protein coding sequences. Inhomogeneous, according to inhomogeneous, and according to the length distribution of protein coding sequences in the genomes, okay, which follows a gamma distribution. So once a gene is emitted, then a transition is made back to the non-coding state, emitting either a emitting non-coding sequence, and then a transition may be made to the gene state on the complementary strand, emitting gene on the complementary strand. Okay. So if a transition is made here, then so this is complementary strand. So a stop codon, and then shadow of gene, okay, and then the start codon, reverse complement. Okay. Okay. So as I said, that these models can be used for generating sequences. So hidden Markov models have been used quite extensively for generating sequences, and now artificial intelligence models are being used for generating biological sequences. You might have heard about generative models like have you heard about ChatGPT? so I think you mentioned last time. So yeah, they have the power, actually these models have the power to generate. So they will learn the parameters and use the parameters to generate the data. So the hidden Markov model trained on bacterial genomes, and here actually the E. coli genome that was used for creating E. coli genome. Okay. So model was trained on E. coli non-coding sequences and E. coli protein coding sequences. Once the model parameters were learned, then that model was used to generate artificial E. coli sequence. And this is becoming really very fascinating these days, because people are now using artificial intelligence models to generate, for example, bacteriophage sequences. Bacteriophage that can mimic natural bacteriophage, but may be more efficient in killing bacteria. For example, antibiotic resistant bacteria, and those bacteria are resistant to drugs. Can we create a bacteriophage that can kill that bacteria because the drug is not working, and whosoever patient is going to die? So you can see the power of artificial intelligence model. It can generate a bacteriophage that can be efficient in attaching to that bug that has become resistant to drug and kill it. Okay. So people are doing research. It's a risky research. US government is quite apprehensive about this, because what happened with coronavirus, generating sequences in the lab, that could be risky also. Okay. But still, I think this is the future people are trying to do, and it's similar actually, generating sequences using models. So artificial E. coli was generated using hidden Markov model, but one gene model was developed. So genes were so there are over 4,000 E. coli genes. They were concatenated, and one model was trained on the concatenated sequences. Okay. So one gene model, and there was one gene model that was trained on all non-coding sequences, and all these were integrated within the framework of hidden Markov model to generate artificial E. coli sequence. Okay. And then what was done was to look at the distribution at third codon position for GC. GC, you understand? GC content, G or C. So that is GC3 distribution. So GC distribution only at third codon position. Okay, so which are shown here for all the genes. So the distribution for all the genes is shown here, and you can see for the genuine or real E. coli, this is this one, and this one is for the artificial E. coli. So what is the difference between real E. coli and artificial E. coli? Artificial was generated by hidden Markov model. What is the difference? Look at the distribution parameters. Mu is mean, sigma is standard deviation. So can you see any difference? Yes. So what is the difference?
Percentage is much higher.
Okay, so you are saying that it has more variability in the GC compared to the artificial. So which is reflected here, mean is more and less same, variability is much less for artificial E. coli compared to real E. coli, right? And this happened because one gene model was made. So, and that gene model actually captured only the major trends among all genes. Okay, so major trends were these. Okay, so most of the genes actually lie in this GC range. So they got captured by the gene model, but those which were at the outliers, so those indicated, for example, in yellow, in sky blue, and here actually purple, they got averaged out. So those signals got averaged out, they were not captured. Only those that represented the majority trend, they got captured by the one model, and this is how it is. In statistics, if you have to model using all the data given to you, then that model will capture only the majority trend in that data. The outliers may not get captured. Okay, so that is reflected here when you generated artificial E. coli. So how to address this problem? Because when you generate it, it doesn't look like a real E. coli. You want to generate a sequence that mimic real, right? Oh, sigma is standard deviation variability. So variability is less for artificial E. coli compared to the genuine E. coli. So in order to address this problem, what you would want to do? Increase the number of models. So you would want to make a model for yellow, you would want to make a model for sky blue, for green, and so on, right? Because if you make one model, it is mostly capturing these genes that have the highest frequency in this range, in the mid range, right? So which are actually blue and green and red, and the outliers are not captured. So to address this, you would want to make distinct models for all these distinct clusters of genes within the genome, and then use those models in the hidden Markov model framework to generate E. coli again. Okay, so so here I am showing you the model using the 3 gene model. So this is hidden Markov model that used 3 gene model, but there could be as many gene models as the number of gene clusters in a genome. Okay, so and then when artificial E. coli genomes were generated again, so you see from 1 model to 3 models. So can you see it is getting better from 3 models to 9 models. So it looks like the model is able to mimic the real E. coli very well as you increase the number of models from 1 to 9, right? So which means that okay, you have to understand the limitations of the model, and then you have to try to address using the resources that you have. So same model, but we increase the number of gene models within that framework, and then we were able to generate E. coli that could mimic genuine E. coli. Okay. So this is the power of hidden Markov generative model, including hidden Markov model, and this is the trend right now. It is happening with language models like ChatGPT, generative model, and this is also happening in the field of biology. These models are being developed for generating biological sequences, and then assessing in laboratory whether these sequences can do what they were hypothesized to do in first place. Okay, if not, then okay, feedback from wet lab, refine the model, change the model, and then check again until you get your desired sequence. Okay. Okay, so hidden Markov model can be used for generating sequences. Hidden Markov model can also be used for prediction. Okay, let's consider a simple example where you have a portion of eukaryotic gene. So eukaryotic gene has exons and okay. So is there some spray something that can, or just water? Not sure if it will work. Oh, it is not working. He's bringing, okay. Yeah, alcohol I think should work. Oh, this may work. Oh. Oh, maybe better. Okay. I can use this, yeah. So, eukaryotic gene. So, typically, eukaryotic gene has coding sequences that are interrupted by non-coding sequences, and coding sequences are called exons. So, let's say that this is exon 1, and then this is intron, and this is exon 2. Okay. So there is a boundary between exon 1 and intron, and that boundary is called 5' splice site. Okay. Okay, this boundary is 5' splice site, and this is 3' splice site. And so let's focus only on this portion. Okay, I want to predict this. So given some sequence, so let's say the sequence is A, C, T, G, A, D, something like this. Okay, and I want to predict this boundary, and this boundary means it's a one nucleotide. Boundary here is just one nucleotide. Okay. So one nucleotide, and to the left of that boundary will be exon, to the right of that would be intron. Okay. Okay. So which is shown here. So here it is. So the model for this is shown. So only consider this portion. Okay. So the model is shown. So a start state. So these exon, intron, 5', they are hidden states. They have to be predicted. Okay, you don't know. What you know is the sequence of what is known to you, sequence of nucleotides. Okay, so you are given the sequence of nucleotides, which is this, and you are given the model. And let's see what the model is, and then you have to predict the splice site, the 5' splice site. And let's see what is the model. You are given the sequence and the model. You remember the model? Probability of transitioning between states. Okay. So probability of transitioning from E to E is 90%. Probability of transitioning from E to 5' splice site is 10%. Probability of transitioning from 5' to intron state is 100%, 1. probability of transitioning from intron state to intron state is 90%, and then transitioning to the end state is 10%. Okay, and rest of there are 3 states. Forget about a start and end state. Other than these two, there are 3 states, and those states emit symbols. Start and end states do not emit symbols. Okay, so there are 3 states. So how many total transition probability parameters? 9. So, but those that have a value 0, they are not shown. Okay, only those that with non 0 values are shown. Okay, so and then so these are the transition probability parameter values. So output probability values. So probability of A, C, G, and T from exonic state, and these all have values 0.25. This is a toy model. Okay, so to just help you understand, they may not be natural actually, these values. Probability of A, C, G, T from 5 prime. Okay, if you look at these values, you will tell me which nucleotide could be the boundary, could be the 5 prime splice site. G, because G has 95% probability, and A with 5%. The splice site will not have C or T. Okay, good. And the probability distribution from the I state. Okay, and the probability of starting in 5 is initial probability. The probability of starting in 5 is, sorry, the probability of starting in E is 1. Right? You always have to start and then make a transition with probability 1 to E. So you always start in E. Okay? So this defines the parameters of the hidden Markov model. So given this and the observation sequence, you have to predict the 5 prime splice site. Okay? So basically you have to predict the hidden sequence. If you have predicted the hidden sequence, and hidden sequence consists of the hidden states, exonic, which is E, 5 prime splice site 5, intronic, which is I. Okay, these 3 levels only. So basically you have to predict the hidden sequence. Okay, so predict probability of hidden sequence given, what is given to you? Observation sequence and model, right? Okay, so you are given these and you have to predict the hidden sequence, and let's say that this probability looks like this actually. So probability of hidden sequence given observation, and I am going to omit M actually just for simplicity. Okay, probability of hidden sequence given observation sequence, and here is S. Okay, and it looks like this. So I am going to consider all hidden sequences, and I will choose one that gives the highest probability of hidden sequence given observation sequence. And I will predict that sequence is the one that underlies the observation sequence. Okay, so I have to maximize this with respect to S and identify one that has the highest value. Okay, this one. Okay, so the problem is actually maximization of this with respect to hidden sequence. Okay, you can write as this. You can write as, tell me, this is probability of S O divided by probability of O. Okay, so omit M. I don't want you to confuse you with so many. Okay, this. So maximization with respect to S, there is no S here. So basically you are maximizing this one, right? This one. And you just did actually, you computed the joint probability of hidden sequence and observation sequence. Okay, so you compute the joint probability of hidden sequence Again I will show you. You did it. Let's go back and see. So actually it is in the next. No, I have not. Okay. So it is here actually. You here it is. You did it, right? We did together. So this is what you have to do. You have to compute this for each of the hidden sequences. Okay. And once you have computed for all hidden sequences, then pick the hidden sequence that has the highest value of probability among all. Okay. Okay, so let's then go back and we'll compute the joint probability. Okay, here it is. So we'll compute the joint probability for each of the hidden paths, and let's assume here that we will consider only those sites as 5' spliced site that has G. Okay, because the probability of being a spliced site is 95% for G. Okay. So I am going to, for simplicity, I am only going to consider those sites that have G. That could be potentially a splice site. Okay. So let's look at those. So where is G? This one. Okay. So this is one of the. So this one is this. So this is a splice site. To the left of it would be exonic nucleotides. To the right of this would be intronic nucleotides. So this one I got this one, and another G, which is this one. Okay. And then G, this one, G, this one, and then this one, and finally this one. Okay. So how many hidden sequences? How many total? 1, 2, 3, 4, 5, 6. Okay. Very straightforward. To the left of the splice site, 5' splice site is exon. To the right is intron. So there are total 6 hidden sequences, and out of 6 we will have to select one, and that one should have the highest probability of hidden sequence and observation sequence, right? So did I write somewhere? I have not written, but I think I wrote here. Okay. Basically maximizing hidden sequence given observation sequence is same as maximizing the joint probability of hidden sequence and observation sequence. Okay. Okay. So now you know how to compute given a hidden sequence. Observation sequence is given already to you. So considering all these 6 hidden sequences, compute this and then select the one that has the highest probability. Okay. And this was done, and it was found. So this is log probability, and maximizing probability is same as maximizing log probability. Okay. So which has the highest value among them? This one, right? This is negative. So this one is the highest. So this one is going to be predicted as the hidden sequence that underlies this observation sequence. And if this is the prediction, where is the splice site? This is the splice site. Okay. So this is how hidden Markov model can help in predicting. So the idea is to find the most likely path that underlies the observation sequence, and most likely path means the path that has the highest probability of being hidden sequence, okay, given the observation sequence and the model. Clear? So this approach is addressed because this is computationally very demanding, using a dynamic programming algorithm called Viterbi algorithm. VITERBI. Viterbi algorithm does this. It computes the probabilities. It identifies the hidden path that has the highest likelihood among all hidden paths. Okay. The other approach is to compute for each position the probability, the posterior probability of the 3 states. So for each position, the posterior probability of being exonic, being 5' splice site, being intronic. So let's say that at first position, the probability of being exonic is 90%, probability of being 5' splice site is 0, and probability of being intronic is 10%, then which label of these three will you use to label the first nucleotide? So, the labels are 5, E, I. E, because the probability of being is 90%. So, you are going to use the label that has the highest probability. Okay? So, this is how for each position you are going to label each nucleotide. Okay? So, this approach is called posterior decoding. You compute the posterior probabilities for each state, okay, and then select the label that has the highest posterior probability. This posterior decoding approach is implemented using a dynamic programming algorithm which is called forward backward algorithm. Again, I am not going to go through all these complicated algorithms, but just wanted to let you know. You should understand the concepts, but there are computational algorithms that can help in predicting the most likely hidden sequence or the states, most likely states at each of the positions. For the former, Viterbi algorithm is used. For the latter, forward backward algorithm is used. Okay, clear? Okay. So, prediction, clear? So, you would want to go through these slides again. I want you to understand how machine learning models can help in prediction. Okay? These are the basics. If you understand this, rest of the things are built on this. You should be able to understand the rest. If you don't understand this, you cannot understand the more complex models. Okay? So, go through the slides again and think yourself. I think you will be able to connect the dots. You should be. So, multiple sequence alignment. So, hidden Markov model has also been used for modeling alignment of more than 2 sequences. So, 2 sequences aligned pairwise, more than 2 multiple alignment. Okay? So, hidden Markov model can also help in describing multiple alignment. So, let us look at a very simple model of multiple alignment. So, alignment of these sequences, protein sequences. And when you look at the multiple alignment, you visualize the positions where you see conservations or variability, right. So, if you look at this, there is this position number 6, which is completely conserved, only G residue, right. Only G is there. So, it is completely conserved. On the other hand, there are other positions that have variations. So, there are mutations or substitutions in other positions. Okay. So, you can build a probabilistic model for this multiple alignment, and for that you would want to consider each position. Okay. So, consider position 1 and you want to compute probability of amino acids at this position 1. So, what is the probability of E at position 1? So, how many residues are there at position 1? 1, 2, 3, 4, 5. Okay. And how many times you see E in position 1? 3. So, the probability of E would be 3 by 5. Okay. So, similarly, you will compute probabilities of all the amino acids at each position, and you can keep it in a matrix, okay, in a table. You can keep it, and then given a sequence which is S1, S2, S9, you can compute the probability of this sequence to be a member of this multiple alignment. This is representing a protein family, okay, to be a member of this family by computing the probability of this sequence given this model that you just computed. Okay. So, probability of S given M, and I would assume that this S1 is situated in position 1. It is not codon position, it is position 1 in the multiple alignment. Okay. So, S1 is situated in position 1 in multiple alignment. So, then it would be probability of 0th order model, probability of S1 in position 1 multiplied by probability of S2 in position 2, and so on. Okay. And these values will come from the table that you just generated, right, and then you compute this. Okay. And this is here. probability of S given the multiple alignment model, and you will divide this by probability of this sequence given random model. So, odds score, okay, likelihood score. So, for that you can randomly reshuffle and then compute the probability of this sequence from the random model. Take the logarithm of this is log odds score, or you can say just say a score or whatever and this can help you in predicting whether the sequence is a member of this protein family represented by this multiple alignment or not. Okay, clear? Okay. So here is how hidden Markov model is used for describing multiple alignment of sequences. This is called profile hidden Markov model, and it is very popular in bioinformatics. You might have heard about PFAM models or hidden Markov models for protein families or domains. Okay. So, look at this alignment, and you can see that it has blocks with no gaps, and there are regions with gaps, right? And those sites with no gaps, they are called match states. Okay. So, let us consider only portions that have no gaps and then join them, and for each position you have a match state. Okay. Match state 1 is representing the first column with no gaps. Now, match state 2 is representing the second column with no gaps, and so on. Okay. Match state. Okay. So, B is begin state, then match 1 and so on, and finally match N and then end state. Okay. And now imagine that you have a sequence that has insertions. Okay. So, you got a sequence and there are some insertions, which means that those residues are not represented in the multiple alignment, right? They are not represented in the multiple alignment because that is something that happened in the sequence of your interest. So, you would want to model the insertion. So, probability of transitioning to match state, and then from a certain match state where insertion happened, you make a transition to insertion state, and there could be insertion of multiple residues. So, that is why there is a self transition, okay? Transition from insertion to insertion again, because the insertion here is emitting only one residue. So, self transition to insertion, and then after enough number of nucleotides or residues are emitted from insertion state, a transition is made back to a match state. Okay? Okay. So, this is how insertion is modeled. Deletion, it is possible that the sequence that you are considering has deletions, which means that there are residues which are actually present in the protein family here, but they are missing from the sequence of your interest. You would want to model it. Okay? So, which is modeled here. So, basically what you want to skip the match states because they are not present in the sequence of your interest because they got deleted. Okay? So, suppose that these two are match states and they are absent from the sequence of your interest, they got deleted. You want to skip them. Okay? So, there should be an arrow from this to this, skipping these two. Okay. So, which is actually shown here by these circles, and these are called silence states. They do not emit residues, they are there to skip the residues in the multiple alignment. Okay. So, if you go from this state, match state, to this circle, and then here to this state, you have skipped this match state because this match state is not present in your sequence, that got deleted. Okay. So, now you integrate all these three, the match states, the insertion states and the deletion states. This is what is the hidden Markov model for multiple alignment. Okay? And this is called profile hidden Markov model because multiple alignment of sequences is often called profile. They represent a gene family or a protein family. Okay? And they are modeled using the three states: match state, insertion state shown in diamonds, and deletion states shown in circles by the profile hidden Markov model. Okay. So here again, and I will skip same thing, but it looks more neat, profile hidden Markov model. Okay. And then you can go to PFAM, and there is a tutorial actually that at, either today or tomorrow you will do. PFAM is used to identify a protein family that the sequence of your interest belongs to. Okay, you have got a sequence and you want to know which protein family it belongs to, because that will help determine or infer the function of that sequence that you are interested in, or you want to know which domains are there in the sequence of my interest. Okay, so that will be predicted using profile hidden Markov model. So you can go to PFAM, you can input your sequence, and PFAM will predict the protein family that the sequence belongs to, and also the domains, conserved domains that are present in the sequence. Okay, so this you will do when you are working on tutorial problems. Hidden Markov model has been used for transmembrane protein topology prediction. Transmembrane, you know, transmembrane, it spans the membrane. So let's say that this is the membrane cell, and this is outside of the cell, so outside, and this is inside of the cell. Okay, so transmembrane protein, it spans the membrane, so it goes inside and then come outside. Okay, and you would want to know that which residues are outside, which residues are inside, and which are within the membrane. Okay, so that's called topology of the transmembrane. And as I just mentioned, you will say that okay, there are three states. One is outside, the other is within membrane, and the last one is inside, right? But of course, this has been modeled using many more states. So you can see the states in these squares or rectangles. So within membrane, there is a helix core, okay, helical structure by these residues. And then outside, which is non-cytoplasmic side, you have loop, cap, and similarly within the cytoplasmic side, which is inside, you have again loop. So it will loop around. So these are modeled by loop state, and then globular residue. For example, if you look at this one actually, so cap, so how many, so you can see the cap states. So cap, let's look at this. So how many cap residues? This is how many cap residues? 5. Okay, 5. And then loop around, right? So what is the minimum residue, the minimum length of loop? No, so you can come here and you can go directly to this cap. So it could be one residue, right? You can make a transition directly here also from this. So just what could be the maximum length of the loop? 10, and then you you can go to globular, or you can directly transition to this. So okay, so 10, but so this, and then so, and then how many are here loop? No, 10, total 20, right? So you can transition, so you can transition to globular, or you can directly transition to loop again. Okay, so 10, and then you can transition here, and then 10 again. So total 20. So loop is variable. So it can even have one residue, or it can have 20 residues. So in between this. Okay. So this is all being modeled here. So this you see how these residues, lengths of these residues are being modeled. So modeling variable length. Okay. And then you have the helix core. Helix core is also being modeled here, variable length. So what is the minimum length of helix that is being modeled? Minimum 1, 2, 3, and then you make a transition here, okay, 24, 25. So minimum is 5 and maximum is 25. So between 5 and 25, okay. So and let's say that, okay, we should be, because these are posterior probabilities, so we can sum up these probabilities here within the cytoplasm or inside. So then you can say that this is the probability of being inside, okay, and then probability of within membrane, and then sum all these probabilities, posterior probabilities, and then that would be probability of being outside, okay. So this was used for predicting the protein topology for a transmembrane gluconate permease 3 from E. coli, okay. So you can see actually the posterior probability plot where transmembrane is indicated by green, inside by red, and outside by blue, okay. So let's look at the first few residues, first few residues. So which has the highest probability, transmembrane, inside or outside? No, this the only outside, blue, okay. So which means that the first few residues are outside of the cell, okay. And then what is the next, from outside to transmembrane to within membrane? So from this position to this position, which is I think close to whatever it is, 40 or so, the residues are within the membrane, okay. And then what next? Inside of the cell, and then again within membrane, and it goes in this cycle. Actually the model is confused at some points. For example, if you look here, it is not clear that whether you should say inside or outside, okay, but it got resolved later on. You can see it is well resolved. So there are challenges because this is using forward backward algorithm, and what forward backward algorithm does, what it does quick, it computes the posterior probability of a hidden state, okay. So that is also called posterior decoding, okay. So and this is actually also the same thing, and a new model was developed later on, and the power of hidden Markov model is that it is modular. You can incorporate as many modules or features as you would want to, and it can work wonder, okay. So in this model they included the signal peptide which leaves the transmembrane, but later on it is cleaved and mature transmembrane is obtained, okay. So they included signal peptide also because there was a cross prediction between signal peptide and transmembrane because they have some shared characteristic. So as I told before that in order for model to work, you have to show model everything, then model is well informed and it does good. Yeah, it will not generate spurious prediction if you have shown model as much information as you can. So this is actually the same thing, but it is helping in preventing cross prediction between signal peptide and transmembrane, and this program is called Phobias, which was developed after the TM-HMM for predicting transmembrane protein. What is the time? 1:10. So we will take a break and we will come back at 2.